All right, hi everybody. Well, here, action. All right, hi everybody. This is your Chapter 10 lecture video. Hopefully you can see these slides. This is our patterns of inheritance. Chapter 10 and Chapter 11 start with our Unit 4, or begin our Unit 4. Just make sure you look at those announcements that Unit 4 is not a paper test at the testing center. It is kind of a case study project-based assessment. It's due on December 7th, so you get a couple of weeks with it. That will be available next week for you. All right, so chapter 10, we're taking kind of some of the concepts that we used from our previous unit, thinking about how cells divide, mitosis and meiosis, the products of those cells, especially meiosis, creating these gametes that can then go to be recombined and create new organisms through that sexual reproduction, creating all that variability. So keep those concepts in mind. Keep that big picture in mind, that meiosis is all about creating genetic variability. We've kind of left mitosis behind. We're not really doing that asexual reproduction. This is all about creating new things, because that is how life moves forward. That's how evolution works, is that we have these kind of slight changes, which we can call mutations, that will change our genetic makeup just enough to hopefully push us forward and not kind of draw us back. So genetics explains these inheritance patterns. We're going to talk about Gregor Mendel and his experiments. But essentially, this is just kind of saying this is how we can explain why people look so different, why we act different, why some people are strong in some ways and weak in others. I had a really great doctor who used to, kind of always describe to me that my issues typically be GI things. If there is some kind of stomach bug going around, I'm gonna get it. But I tend to not get upper respiratory infections very much. I don't get skin infections very much. Those things just don't tend to impact me. And so she kind of explained, she was like, yeah, that's kind of your genetic makeup. Everybody has a place that they tend to be a little vulnerable. For me, it's through my GI tract. And so we all have kind of these strengths and weaknesses. I used to live in a neighborhood that had a lot of little baby bunnies. And for years, there were all these little baby bunnies, and they got really comfortable with everybody around them, and there really weren't any predators kind of coming after them. And after about 2 or 3 years, the coyotes and the owls and everything started to move in, and these bunnies that were so trained or so used to in their genes not having to run or be afraid, they got snatched up quick, and all of a sudden we had these really big owls. And all of our bunnies that were so friendly and would just stand in the yard and let you walk past them got really skittish. They started learning how to run faster, and the bunnies that had those genes that adapted them to be more sensitive to movement from the side, from their peripheral vision, those rabbits that were more quick to react and to jump just a little bit faster, whose we'll talk about sodium potassium pumps, whose nervous systems could fire just a millisecond faster, were able to get away from the snake or the owl or the fox or whatever it was. And so that's all genetics. That's what that comes down to. So when you hear about the idea of survival of the fittest, that's what that's referring to. It means that the organism has adapted just enough to their environment that they're able to survive, to reproduce. That's what we kind of are looking for when we talk about fitness, is do you live to reproductive age? Because if you live to reproductive age, that means your genes have been good enough to pass themselves along. And so that's kind of that marker. It's a pretty low marker, but for genetics at least, that's what it's going for. That's the end game, is just passing those genes along, making this organism adapted enough, not perfect, right, but suited enough to be able to live long enough to pass along its genes. So we all have different strengths and weaknesses depending on our environments. All of that information is packed into our chromosomes. This is just a little bit of terminology review, so I'm going to go quick through this. If you remember, we're looking at a karyotype here. Humans have 23 pairs of chromosomes. You have 1 chromosome from mom, 1 chromosome from dad. Again, hopefully those combinations make you well suited to your environment, suited to this day and age, wherever you may live and work and do. All of that is packed into your chromosomes. Each chromosome has a region or a section called a gene, and that gene is going to code typically for a protein. It may be RNA or mRNA or tRNA, something like that, but related to protein synthesis. Our chromosomes are packaged into genes or sectioned off into genes. Each gene is made of a sequence of nucleotides, those A's, T's, C's, and G's, right? So hopefully that's all kind of coming back. Our normal body cells, our somatic cells, are diploid because they carry 2 copies of every chromosome, but our gametes, our egg or sperm, are going to be haploid because they only have half of the genetic material. That'll allows them to recombine in fertilization to create a full complement of our genome that an organism needs, which is why egg and sperm don't just spontaneously grow into organisms. They only have half the material. It's not until that full genetic complement is there that protein synthesis can actually start to take place. Each chromosome is going to, again, like I said, have a section of it that is going to code for a certain gene. Depending on the chromosomes, on the genes that you inherit, you might have the exact same sequence of nucleotides on those genes. So both of your parents, I'm going to simplify a lot of these examples just to kind of keep it straightforward for us. But like, let's say eye color. Eye color is controlled by multiple genes, but let's say it's just one. Okay, so both of your parents have blue eyes. You have the gene, that allele for blue eyes. But if one of your parents has blue eyes and one of them has brown eyes, you have a chance of getting different alleles, different variations of a gene. So that's what an allele is. An allele is a variation of a gene. There could be one allele for a gene. There could be just one variation of it. There could be 24,000, or that's an exaggeration, but there could be, dozens of different variations for a specific gene, different alleles for a gene. It just depends on that specific one. We're going to talk about how we can categorize those different alleles. So Gregor Mendel is the one that we usually associate with kind of the father of modern genetics. He was the one that really took a lot of time to look at how pea plants were passing along their traits, because farmers typically want to have a specific trait that they want to breed for. Farmers have been doing selective breeding. Humans have been doing selective breeding since we started agriculture. Finding a cow or a pig or a sheep that was really docile, really able, easy to keep, very productive. Okay, well, let's take that one and let's just keep breeding it with a lot more. So hopefully all of those offspring have that same trait. That's a very kind of common thing. But then you would notice, oh, well, how is it that sometimes, we have this one standard, and out of 20 sheep, those all 20 match the standard, but then there's a couple that are just outliers. Where do those come from? Why are they like that? This was a question that people have always kind of noticed. Mendel had the time and the resources to be able to look at that. So he studied pea plants. Pea plants, you can look in your textbook, they have a bunch of different traits that are very easy to follow. And so Mendel was lucky in that he picked something that had very predictable traits that inherited their genes in very predictable ways, and were very obvious in that. I say lucky, maybe he was smart. Maybe he kind of noticed what he was doing already, and he was like, hey, I think that this is going to be a simple way to do it. But there was no concept of genetics at this point. They literally just saw the peas, and then the seeds, the plants, and what they produced. That's what they were looking at. They didn't have this idea of this genetics. It wasn't a thing. So offspring was giving Mendel clues about the genes of the parents. And that's what you can always think about, is that the way that you look says something about the genes you got from your parents. Okay, so how you look is what you get from your parents. That's why sometimes your child, your offspring, like if you have a child, maybe your child looks like your parents in ways that you don't. Your child might have traits that your parents have that you didn't really inherit. It can skip generations. You may have heard of that, of certain things skipping generations. It's all because of what you pass on, and then what gets combined during fertilization from your other parent. There's all these interactions that are happening. So there's a couple of different ways that we can look at our development, or our offspring. We can talk about true breeding versus hybrid. A true breeding means that when you fertilize a plant with itself, it's going to produce exactly what it is. All of those offspring are going to be exact to the parent, same copy, very reliable, very predictable. Hybrid, you're going to get this mix where a lot of them are going to look at the parent, but then maybe some of them don't. And there's something in there. It's like, why is that? Why is that this one green seed pops up when all these others are yellow and the parent was yellow? Where's that green coming from? We're going to look at that. A lot of this comes from the fact that we have, with the alleles, dominant and recessive alleles. So again, just because there are dominant alleles and recessive alleles doesn't mean there's only 2 alleles for every gene. You can have 10 different alleles. Maybe 7 of those alleles are dominant and 3 of them are recessive, or maybe it's vice versa, right? Maybe half and half. It can be a different combination. So it really just does depend on the specific gene that you're looking at. Mendel was, again, studying yellow and green. These pea plants only produced yellow and green. They only had tall or short. They only had wrinkled or smooth. So each trait he was looking at only had 2 options, and they were very clearly dominant or recessive. So very, very straightforward, what we call Mendelian genetics, Mendelian inheritance. So some alleles are dominant. That means that when they are present, they are always going to produce, always going to be present, always going to be expressed is the term that we'd like to use. So if you have a dominant allele for a gene, if that is your genotype is to have a dominant allele, you're going to see that. And a lot of times, especially in plant seeds at least, it's because it makes the protein that makes the yellow. That's what's happening here. Green is the recessive. The recessive allele is one that is not going to be expressed. You're not going to see it. And so for pea plants, it's actually a non-functioning protein. The enzyme that this recessive allele codes for just is non-functional protein. And so you get this green color instead because the enzyme doesn't work. So a recessive usually, again, are going to be encoding for non-functional proteins. This doesn't mean that recessive are less common or more common. That's something that I always like to make sure that we're highlighting. Dominant doesn't mean more common. A dominant trait could be much less common in nature and in society, but it does mean that if a dominant allele is there, if one of your chromosomes has a dominant allele, you're going to show that trait. So looking at the offspring can determine, again, the genotype of the parent. Individuals with the same phenotype can have different genotypes. So you can have, for example, here, yellow seeds. Well, this yellow plant is producing all yellow seeds. This yellow plant is producing some yellow and some green. So that should tell you something about the genotype of this yellow parent here, that if they are not producing all yellow seeds, or they're producing some green seeds, something's not quite right. And so what that means is that they're probably homozygous or heterozygous. We have to start looking at these kind of terms now. So this, again, refers to the types of alleles that you have. So if you have the same two alleles for a trait, you're considered homozygous for that trait. Homo meaning the same, and zygous referring to the zygote. When sperm and egg come together, that single cell that's restored the genetic the chromosome number, that's called a zygote. And so homozygous means that that cell has the same alleles for that trait. If you are heterozygous, you have different, hetero being the opposite, or not opposite, you have different. Heterozygous, you have different alleles for that trait. So for this, if you're heterozygous, you're going to have one dominant and one recessive. That's just typically how it is for these inheritance patterns. If you're homozygous, you always have to clarify if they're homozygous dominant or homozygous recessive. If the individual is homozygous dominant, they're going to have the two dominant alleles for this trait. If they're homozygous recessive, they're going to have two recessive alleles for this trait, and they can only pass along that allele. A homozygous recessive chromosome or gamete is only going to be able to pass along, or individual rather, is only going to be able to pass along a recessive trait. A dominant individual will only be able to pass along this dominant allele. Heterozygous can pass along each one, which is why you have a parent here who is displaying the dominant yellow color, actually producing green and yellow. Because yellow, you can have the yellow phenotype, the yellow expression, but actually have the heterozygous still carry the recessive allele in your genotype. What's not possible is to have a recessive presentation, a recessive phenotype, and then produce a dominant offspring. That won't be possible unless the parent in a self-breeding capacity, in this capacity at least. If you are mixing with another parent who could produce a different allele, dominant allele, then yeah, sure. But in terms of this green seed here being able to pass along a dominant allele, it's impossible, because if you're green, you have to be homozygous recessive. If you're yellow, if you're presenting as yellow, you could be homozygous dominant, or you could be heterozygous. You don't really know until you take those yellow seeds, you start planting them, you start reproducing with them, and you see, well, what do they actually make? If I plant these seeds, if I grow these seeds to fruit, what are they going to produce? Are they going to produce all yellow, like here, or are they going to be hybrid and actually end up being heterozygous? So again, just kind of summarizing seed color, it's a pigment coating enzyme because genes are going to be coding for proteins, enzymes are proteins. And so the dominant allele Y, and the letters really don't matter. They're specific if you're given a letter for an allele, that's the letter you should use. But for the most part, they don't really mean much. They're just kind of a placeholder for us. Y is yellow, that's the dominant trait, so they pick that letter for the allele. So the dominant allele Y is going to make an active enzyme, a protein that actually folds into a functional functional enzyme that can convert and create that yellow color, whereas the recessive is a non-functional protein and it doesn't make any of that color. So one of the ways that we can start to do this, because again, Mendel was, he was having to do this. He was having to take seeds. He was having to plant them. He was having to then grow them all the way until they started to fruit. And then he was having to look at those seeds. And that was how he was able to kind of determine if a plant was homozygous or heterozygous, right? Because if you have a green seed, you know immediately, oh, this green seed is going to be heterozygous, is going to be homozygous recessive. It is only going to produce recessive alleles. If I take a green seed and mix it with a green seed, I'm only going to get green seeds, green peas, right, whatever. But if you have a yellow pea, you don't know until you start doing enough reproductions, until you go through enough reproductive cycles, you don't know if that yellow pea is homozygous dominant and always going to give you yellow, or if it's actually heterozygous and is going to give you some green ones in there. Because some farmers might not like green peas. Maybe they want all yellow peas, right? Some people are very particular about that. Children specifically. All right, so Punnett squares are a way that we can help to do this. They represent gamete formation, fertilization, so they are predictors. They allow us to kind of look at what we have, look at our phenotypes, predict our genotypes, or take our genotypes, and then make predictions about how frequently a trait is going to be coming up in a population. It's important to know that it's not exact. So Punnett squares typically, in a normal ratio, we're going to kind of give you a 1 to 2 to 1 ratio of homozygous dominant, 1, 2 heterozygous, and then 1 homozygous recessive. So it's a 25% chance of having both homozygous dominant or homozygous recessive, and then 50% chance of having a heterozygous individual. These ratios only show up in large populations. So when we're talking about human reproduction, we only tend to have like 1 to 4 kids is average, I don't know, somewhere around there. And so those numbers are small enough that these ratios don't always play out perfectly. So if you're kind of trying to apply ratios directly to human reproduction, doesn't always work that way. So Punnett squares, a Punnett square is this square that you see here, and then we put the parents at the top. So female parents, female gametes at the top, male gametes over at the side, and then you just cross multiply for them. Remember, every parent is going to have 2 alleles. Every body, for every trait, you have 2 alleles for every trait. Those could be the same alleles, or they could be different alleles. And so it really just depends on your phenotype. Depending on your phenotype, you might be able to directly determine, oh, I know my alleles. I am a green pea, I know my alleles. If I'm a yellow pea, oh, I might not know my alleles. Depending on my parents, depending on my offspring, I can start to predict those things. I can start to kind of work backwards and figure that out. So fun genetics puzzles. The Punnett square allows you, if you kind of have the parent gametes, you can start to predict it out and start to look at what those predictions would be. So in this case, you have 2 yellow parents that both happen to be heterozygous for that trait. So they have the dominant and recessive alleles. So you'd write the dominant and recessive, the kind of 2 up there, and then just do your multiplication out. This one went ahead and made the color coded for you as well, so you can see that even though only one of them is homozygous dominant, they're still all 3 presenting as yellow because they have at least one dominant allele, and they're going to look yellow. This last one only got both of the recessive alleles, so it's going to present as green. And so we have a genotypic and phenotypic ratio. Your genotypic ratio is reflective of homozygous dominant, heterozygous, or homozygous recessive. So you have a 1 to 2 to 1. 1 is homozygous dominant, 2 are heterozygous, 1 is homozygous recessive. That's your genotypic. Your phenotypic is 3 to 1 because you have 3 yellow seeds and 1 green seed. So that's the difference from that phenotypic and genotypic. You can present that as percentages as well, 75 and 25. That's very normal. So in this one, again, we have this heterozygous parent male also crossed with a heterozygous female. We call this a monohybrid cross because you're crossing 1 trait. We're only doing 1 trait at a time. And truly, if you get good with Punnett squares, you kind of can hold it in your brain. If you know that both of your parents are heterozygous and they're both are going to look the same, this is always going to be your ratio. This is always going to be what it is. So for some of the Unit 4 questions at assessment, some of them say show your work. Show your work means that you have to show me the Punnett square. If you don't show the Punnett square, you don't get the point, even if the question is correct. Some of them don't say show your work, and you can just write it out. Do the Punnett square if you want to, but if you don't need to, you don't have to. Just be careful for that. Look for that and make sure that you're answering that question fully. Okay, so Punnett squares, like I said, are giving you these ratios out that we can then use to start predicting what the offspring might look like. And so it's a way for, again, in this case, for farmers to be able to say, okay, well, if I plant these seeds, this many are going to be yellow, this many are going to be green, so I can kind of sell based on that ratio. If you are doing genetic testing, you're two parents that are trying to have a child, and you want to know what are the risks, what are the chances of our child having this condition based off of our genotypes. You can use it for that purpose as well. So Punnett squares, again, help us to answer some of Mendel's questions. One of those being, why is it that if yellow is dominant, why do some yellow seeds produce a green seed instead? And again, it comes down to this fact that you can have a heterozygous individual where your phenotype is presenting as a dominant phenotype, but it's because you only need one dominant allele to fully present. You can have just one dominant allele makes enough protein. You don't need both alleles to do that. We're going to see that with some traits, that's not true. Some traits you do need, if you have both dominant alleles, you actually show more of it. If you only have one dominant allele, you don't show as much. Sickle cell anemia is a condition like that. If you are heterozygous, you actually don't show the condition as much as if you were homozygous dominant for the condition, or homozygous recessive for the condition. You actually have a more mild case of it. And so there's some examples, plenty of examples of like that throughout genetics. But in this case with seeds, because they are completely dominant, we're going to talk about incomplete dominance, codominance, other types of inheritance patterns. But with seed color, seed shape, plant height, these were all dominant traits for pea plants. And so it was very easy for him to track. Easy relatively, I don't want to discount his work. Okay, so Punnett squares, again, summarize meiosis and fertilization, this idea that, with meiosis, it is creating genetic variability. You're taking a gamete, or you're taking a cell, you are splitting its genetic material in part. You're actually scrambling some of that a little bit during crossing over. We're scrambling some of that genetic material, pulling the sister chromatids apart, taking these homologous pairs and separating them out, and then combining them with someone else, taking sperm and egg and putting them together to create two totally different and in a completely new novel way. What is it? Something like 8 trillion different combinations or something for each of us. We are 1 in 8 trillion chance of you being you, which is very cool. Unless you're a twin, in which case I'm so sorry. I guess that's 1 in 4 trillion. Sorry, that sounded bad. All right, so Punnett squares, again, summarizing meiosis and fertilization. You have this segregation of alleles, again, taking apart the homologous chromosomes, splitting up the sister chromatids, and then recombining them with the sister chromatid from a chromosome from another individual totally. Gives you all these new kind of alleles. Maybe you get 2 dominant alleles, the same ones. Maybe you get a dominant recessive. Maybe you get 2 recessive, right? It's this kind of interesting combination, which is why you kind of look like your mom. You kind of look like your dad. You maybe have some traits from your grandparents or from an uncle that you share, right? Those kind of random little things where it's like, well, you kind of have that. It's because a lot of our traits aren't just controlled by one gene. We're going to see that some traits are controlled by multiple genes. So this one just kind of hitting home with some of these human traits. Like I was talking about, some parents might go through genetic testing just to see. So in this case, we're looking at cystic fibrosis. Cystic fibrosis is a condition that essentially causes malformed chloride channels in your cells. So chloride is really essential for moving molecules and ions back and forth. And so with people with cystic fibrosis, in their lungs, chloride is used to help move mucus around. And so when they don't have that, mucus tends to accumulate. And so this is one of those genetic conditions where it is a recessive condition. And so if you are a carrier for it, you can be what we call a healthy carrier. So a carrier for a condition is when the condition is recessive. So you have to have 2 recessive alleles for it, which means that if you have 1 dominant allele, you're not going to show the condition at all. 1 dominant allele is enough to make that protein and you be perfectly fine. However, you're a carrier for it. So if your partner is also a carrier for it, there's a 25% chance that your child would be affected. There is a 50% chance that your children would be healthy but carry the disease as well. And there's a 25% chance as well that they would be completely unaffected. They would be healthy non-carrier. And so we have to, with genetic diseases, we have to kind of parse out, okay, well, are you healthy non-carrier, healthy carrier, or are you affected by this? And that is going to change depending on the condition being the dominant or recessive, that kind of ratio. So always just kind of being aware. And for the most part, I will always tell you if a condition is dominant or recessive, or I will give you the inheritance patterns, and you should be able to figure it out from there. I won't ask you to just memorize in your head and just remember that cystic fibrosis is recessive. It's good to do. It's helpful to have that in your brain. But I'm not going to ask you just to memorize which conditions are dominant, which ones are recessive. I would always give you enough to get you there. So with this, we've kind of been looking at individual little traits in these monohybrid crosses, taking one trait at a time. But sometimes we look at multiple traits. Sometimes it's helpful to look at a couple of things. So we can do a dihybrid cross, where in this case, we're looking at both seed color and seed shape. So round versus wrinkled. Again, these pea plants were pretty straightforward. So they could be green or yellow. They could have round seeds or wrinkled seeds. And so let's say that you are a person who particularly wants to have green wrinkled seeds. That is the exact seed that you want, and you want all of the pea plants that you produce to always be green wrinkled seeds, because that to you is the best flavored seed. Okay, well, green is recessive, so you need to make sure that you're getting a recessive seed, and then wrinkled is also recessive. And so for that, you have to have what we call a double recessive seed. Little r, little r, little y. Little y is going to give you this green wrinkled seed every time. Yellow and smooth, yellow and rounded, those are the dominant traits. And so that's the ones that if those alleles are in there at all, they're going to present themselves. And so that's why you see fewer green seeds and fewer wrinkled seeds than round seeds. Again, with complete dominance, tends to be the case. It can be the case, but it's not always true that dominant is more common. I have to keep reminding that because in here it looks like that. But again, we'll see some diseases like Huntington's disease. Huntington's disease is a genetic condition that is a dominant condition in humans, and it is fatal. It's typically fatal by 30 or so, but you don't see it. It's not that you don't see it. It is not a very common condition because it is so fatal. It tends to not spread very, very rapidly. So with dihybrid crosses, we're looking at two different traits here, color and shape. It can start to get kind of confusing, and it isn't always possible to inherit these, to predict these inheritance patterns. It really depends on the actual location of the gene. So if you kind of remember, we have to kind of go back to some of these ideas of what happens in meiosis. Things like independent assortment. During independent assortment, your homologous pairs can line up on either side. You can have kind of a different orientation depending on which way you want them to be, or different offspring, different gametes produced, depending on which way your homologous pairs of chromosomes oriented themselves. And again, it gets doubled because you have your egg and your sperm both going through that independent assortment. And so this independent assortment is starting to talk about separating traits. And so in some conditions, some of your traits might not be inherited together quite as smoothly. And so that's the idea that you can have this kind of mixing where it's like sometimes you get green and wrinkled, and sometimes you get green and round. Sometimes you get yellow wrinkled, and sometimes you get yellow round. It's not always going to be green wrinkled and yellow round. It can mix and match. It's a long way to get there. So it can get really confusing, especially if you're trying to look at multiple traits. Let's say you want tall plants that are green and wrinkled. Okay, well, now we just threw something else in there. We just threw a third trait in there. Instead of having to do a full tri-hybrid cross, what you can do with these Mendelian traits where it's very kind of complete dominance is that you can just take the percentage, the likelihood for one trait, for each trait, and multiply them by each other. So let's say that you wanted, let's say you want your farmer, in this case, this farmer, this person wants a variety of seeds. So they want to make sure that they're going to have all heterozygous offspring. They want to have round and wrinkled. They want to have green and yellow seeds. They want to have tall and short plants. They love the variety. And so what you do is you kind of take your Punnett squares for each of those traits, seed shape, seed color, and plant height. And you say, okay, well, if this person wants the heterozygous, for each of these traits, it happens to be the same. So it's all 50, it's 1/2, 50%, 0.5, whatever number, however you like to look at numbers. You just multiply them together, right? Half by half by half. Oh, and there's a 1 in 8 chance that, 1/8 chance that we can get full heterozygous offspring for this person. So that's a pretty tough call. Maybe not so much, considering that you're looking at 3 different traits, but still, it's going to be pretty tricky. So you can charge more for that. I used to work with horses and train horses, and my trainer always said, you know, if you can find 2 horses that are good, don't be as particular as you can. Be a little less specific, because as soon as someone knows you're looking for something that's very specific, very minute, then they can charge you more for it, because then they might have that. It's harder to find specifics. So this only works for genes that are close together physically on the chromosome, genes that we call linked genes. And so sometimes that does happen, where you are going to always have things that are kind of inherited together, these kind of 2 traits that kind of get inherited together. So when they are physically close together on a chromosome, it's less likely for them to get separated during things like independent assortment and crossing over. Remember, crossing over happens during prophase, when they actually, the 2 homologous pairs, sister chromatids from homologous pairs, actually change genetic material. So the chromosome from your mom that was originally your kind of parental chromosome, oh, why am I talking? Here we go. I was like, wow, it was right there on the next slide. So you have during the crossing over in prophase 1, you have your parental chromatids, so dad's and mom's kind of parental chromatids, but then they're carrying just this extra bit of genetic material. So again, this is why you tend to look like a combination of your parents. So this is only true for genes that are physically close to each other. In this case right here, you can look at a linkage map. And so a linkage map is essentially a way that you can kind of look at, in this case, they were looking at fruit flies. Fruit flies, scientists love to use those. They have very kind of specific traits that we can look at. And so with this one, I believe, I can't remember exactly what the traits are, but things like the wing veins, the eye color, the placement of the antenna, the mouth parts, all of those things can kind of be observed. And when you start to observe that, you can see, oh, well, these 2 things are always going to be the same. If you're going to have this one trait, you always have this trait as well. And so that typically means that they're going to be close together. Whereas some traits, it's like, oh yeah, you can have, the yellowish color to your body, but you're never going to, but you may or may not have this specific wing vein. And so those 2 things probably aren't related to each other. They're probably not very close, because you rarely see them always together. Some traits are always inherited together, and that means that they're going to be very close on that on the chromosome physically. It's always good to remember when we start thinking get into genetics, that we're talking about physical things. These are not just kind of esoteric molecules that are using some, I'm in like a space background, some kind of, unseen mystical force. No, these are like physical interactions that are happening between these things. There are physical places on this chromosome where these things are, and they can get kind of stuck with each other. They get stuck together. So again, a lot of this, the independent assortment, crossing over, complete dominance, this is a very Mendelian traits, Mendelian genetics. But not everything follows Mendel's traits. Some follow very different inheritance patterns, and a lot of traits follow very different inheritance patterns, which is why genetic conditions can be very hard to study, predict, and treat. So in this case, we're looking still at flowers. We're still looking at plants. So don't think that plants all use complete dominance Mendelian patterns. Here you see one plant that has red flowers, one that has white flowers, and their offspring ends up being pink flowers. So this is kind of a different type of inheritance. This is called incomplete dominance. So it's where you see a blending of the traits, a blending of the parents. With this, when we write out the alleles for incomplete dominant traits, they picked R, so which kind of does, maybe it stands for rose, maybe it stands for red, I'm not sure. But rather than using a capital letter, a capital R, they use lowercase letters and then add a number. So R1 stands for the actual red color. R2 is the white color, and then R1, R2 is this blended kind of pink color. If a plant in this case is a pink color, if it's showing this blend of the characteristics, it's actually R1, R2 is its genotype. If it is fully red, it has to be R1, R1 because it's not diluted. If it's fully white, it's R2, R2 because it has no pink shading in it. But again, if it's pink, then it's a heterozygous offspring. Again, this is because each of these, R1 and R2, make proteins, but they don't really make enough to mask each other, and they just kind of blend together, right? What's interesting about this is that if you take red flowers, white flowers, you make them together, you get your pink flowers. And let's say you take this, you take two pink flowers. If you're pink, you have to be heterozygous. So we have 2 pink flowers. They're both heterozygous, R1 and R2. If you make them together, you're going to get red and white flowers again. you're actually going to bring that trait back out. Most of your plants are going to be R1, R2. Most of your plants are going to be heterozygous and present as pink flowers, but you're going to have some that are homozygous red, homozygous white, and you can bring that trait back out. You can breed it back out from them, which I always think very interesting. I'm sure kind of surprising if you're not used to working with flowers, thinking that you have all pink and then you go to plant those, and all of a sudden the next season some red ones and some white ones pop back up. It's like, wait a second, I bought pink. How did this happen? And those pink ones will still be there. So this is incomplete dominance. You have this blending of traits, this blending of characteristics. Co-dominance is when each one is going to be presenting itself fully. There is no blending, there's no masking, so it's not complete dominance, not incomplete dominance, it's a co-dominant trait. So every allele is going to be presenting itself. That's functional. So blood typing is a common example that we use for this. The common types, the blood types that we typically see and that we associate that are relevant are type A, type B, AB, and type O. So 4 blood types: A, B, AB, and O. Those refer to the proteins on your cell surface. We call them antigens. Antigens are on your cell surface, and they are kind of the protein markers like that say, hey, this is a body cell, this is the kind of cell that I am. So if you're type A, if you have type A blood, that means that you have this A protein, which obviously doesn't look like an A. That's just the letter we associate with it. Okay, so you have this type A protein on your cell surface. If you're type B, it's B protein. If you're type AB, you don't have some kind of blended protein that's C. No, you have the A protein and the B protein. 2 totally different ones are both on your cell surface. They're co-dominant. One isn't going to mask or hide the other. They're not going to blend together into a new protein. They're going to make 2 totally different proteins that are both on your cell surface. So these are the dominant alleles. A and B are both dominant alleles, and so they present together as co-dominant. There is a recessive allele, this little lowercase I that you're seeing. It's only present in type A, type B, and type O. If you are AB, you have to be heterozygous, IA, IB. If you are type O, that means that you actually don't have any proteins on your red blood cell surface. There's no, none of these markers are on your cell surface. And so the O stands for kind of nothing. And so in that case, you are recessive. You have the recessive alleles, which are these little I's. And again, recessive alleles typically are non-functional proteins. And so if you have this recessive allele, your proteins don't fold into a right shape. They can't actually stick to the cell surface. The I stands for immunoglobulin. It's a red blood cell thing, right? It's an antibody thing. So co-dominant alleles, they don't mask each other. They're both presenting fully. If you, for blood types, for the genotypes for blood, we use IA. Sometimes you will just see A, B, AB for the genotype, but it's also very, very typical to see IA, IA, or IA little I. If you are heterozygous for A, you have IA little I, you're still only going to be type A. If you are heterozygous for B, IB little I, you're still only going to be B. If you are type AB, you have to have IA, IB as your genotype. That's what you got to be. If you're type O, you have to be little I, little I. You can't have anything else. That's just how that works. So this is, again, just kind of some non-Mendelian traits. We had our Mendelian traits where it's very true, complete, incomplete. You have a dominant and recessive. You have complete dominance where you have a dominant and a recessive allele. Incomplete dominance where we see this blending. And then this co-dominance where each one is presented fully. Some of the things that can happen, we call it pleiotropy. So this time, this is one of those things where you have one gene that actually has multiple effects. It's actually going to have many phenotypes. So for this one, we're looking at Marfan syndrome. In Marfan syndrome, you have this non-functional protein that actually then goes on to affect your eyes, your bones, and your joints, and your heart and lungs. So it's just kind of amplifying the effect. It can be one gene that does many, many things because typically it's a starting product, right? It's kind of causes some defective connective tissue that then is going to show up everywhere else. So if this one thing doesn't work, none of that works. Similar thing with epistasis. Epistasis is when you have to have one protein working for the other one. So blood typing is an example of that. For proper blood typing, you have to have this one protein that actually attaches the identifying protein to the cell surface. So if you don't have a functional protein there, you're always going to show up as type O blood, even if your supposed to be type A. You have the genotype for type A or type B or AB. If you don't have this first protein marker, nothing. Some other things start to happen, like with sex-linked traits. Most of the traits that we were looking at, these were all non-sex linked, so they were in our autosomal chromosomes, those first 22 pairs of chromosomes. The last two, we start to see some different traits just because of the fact that males and females have different sex chromosomes. Males are XY, females are XX. Because of this, when we are talking about sex-linked traits, we have to make sure that we are looking at, or we're kind of asking about, are we asking about a male or a female? Because it's going to be different. Part of the reason is because, if you're a female, you have two X's. Well, if a trait is, let's say, recessive, men only have the one X chromosome, so they're always going to show that trait. Whereas females, if you have a recessive trait, well, maybe your other X has the dominant allele, and so that will be produced. So because males only have this one X, whichever allele is showing up on that X chromosome, that's the one that's going to be expressed in their phenotype. Offspring, so this is in fruit flies looking at their eye colors. So eye color is associated on the sex chromosomes in fruit flies. And so in this one, we see a female, heterozygous female XR, which is that dominant red, and X little r, which is the non-dominant, I believe, the white. So for females, all they need is the one dominant XR. So you're going to almost always see that. You'll see that a lot, very often. That if you have a female and a male with red eyes, the male only has the one X, so he's going to have to donate that. So if you have, in this case, a male with the dominant trait, that's the only X that the male can donate, and so the female, his daughters, are always going to show that dominant trait. It's one of those things that you have to, again, kind of keep in your brain, that a male is going to have XY as the gametes that it can donate. And so if you're talking about an X-linked trait, the male can only give that X. So whatever the male has, that's what he's donating. If the male has a dominant X, he's going to give his daughter the dominant X. Females have two X's, so they're going to always donate an X to their son. They can't donate a Y. The sperm is the gamete that determines sex in offspring, because sperm can donate an X allele or an X gamete or a Y, and so sperm are what determine your offspring. So if you're looking at the sons of the sons of the cross, the Sons of the Cross sounds like an anarchy group. If you're looking at the Sons of the Punnett Square, your mother is donating your X, and so if she has a dominant allele, that's what you're getting. If she has a recessive allele, that's what you get, no matter what. For the female, right? If your mom is donating your X, if she's recessive, you also have to get a recessive allele from your father to show that trait. So it's just one of those things where X-linked disorders typically affect males more than females. So your X-linked recessive disorders, you're typically going to see them in men more than women, just because men only have that one X. Common one here is hemophilia. Hemophilia is a blood clotting disorder where your blood tends to not clot properly and just keeps bleeding. And so again, this goes back to our healthy versus carrier versus affected. With this cross, you're having a heterozygous female, and then you're having a healthy male. So we say healthy male because his X is the dominant allele. He is healthy. He does not have hemophilia. He's going to pass a dominant allele to his children or his daughters. His daughters are not going to be affected because he's got that one dominant allele. That's all that they need to be non-affected. However, he's only passing his Y to his sons. He's not giving his X to his sons. So it's up to the mother. Whatever allele the mother gives to her son, that's the one that's going to be expressed. So she'll have the chance to have one healthy male, but then also a male with hemophilia. Here's just a table of some of those conditions. Again, I'm not going to ask you to memorize which ones are dominant, which ones are recessive, but I will give you information. You should be able to figure that out based on the information that's presented to you. So this is just one of those things that's kind of interesting to look through the different X-linked recessive or dominant disorders, what their genetic explanation is, how they kind of occur, like what mutation might occur to make them happen, and then as well what the characteristic is. Just because you'll probably see as you go through biology a lot of these conditions coming back up, and so it's nice to just kind of have them in your brain and just to be familiar with them. So one thing that tends to happen with females, because like we said, males only get the one X, and so they're going to show that trait, whatever that X is, that's what they show. With females, because they do have two X's, if they were always constantly showing both of those traits, it could get very problematic. And so typically what happens, what actually happens in females is what's called X inactivation. So every cell in a woman is going to have only one X chromosome that's actually making its proteins. The other one is just going to be turned off. It's not going to be translated. And it's completely random. One cell to the next, its neighboring cell. One cell might have one X chromosome turned on from the dad. The next one, its neighbor, is going to be from the mom. However, what is interesting is that whatever cells, let's say if you have a cell that has your X chromosome from your dad turned on, all cells produced, all those mitotic divisions are always going to have your dad's X chromosome activated, not the mom's. So that does maintain consistency throughout the genetic line. X inactivation is what explains calico cats. So calico cats are always genetically female because you need to have two X's. You need to be able to have one X that you can turn on, one X that you can turn off, and it needs to be this random pattern, which is what gives you this kind of random banding pattern. Calico cats are always orange, black, and white. You got to have all three of those colors. And so it's this random activation of X's. I will say there is truly one example. It is possible to be XXY. That's a genetic condition where you have two X's and a Y. So you're male technically, but because you have two X's, you could have this X inactivation pattern. It's very, very rare to see. X-linked dominant disorders, again, are also less severe in females because of that X inactivation. So some of the X's that carry the dominant disorder, they will, produce their proteins, but because in some of the other cells that will be turned off, it may be less severe. So it really just depends. If most cells are expressing your unaffected X, then you have a more mild condition. If more of them are expressing it, it can give you to be more severe. Depending on the condition, if it is dominant and it is fatal, then males do not live to, this is a non-viable embryo just because they only have that one X that can express that trait. And so they just won't be able to survive, even if because they only have the one X. Environment can also alter phenotype. So in this case, we're looking at a Siamese cat. Siamese cats typically have this very common white body and then this darker tail, feet, face, ears. And it's because these are colder. They have less blood supply going to this area, or rather they're losing more heat in these areas. And so that temperature actually causes their cells to express a darker protein versus the body, the central body, which has a higher temperature, expresses this more white part. And so the color is not produced there. This is called epigenetics. So the environment can alter your phenotype. This is one of those things where if you know any identical twins, if you know identical, I met a set of identical twins when I was riding the bus in middle school, and I still know them. They still kind of live around me. And when we were growing up, they looked completely identical, couldn't tell them apart. But as we got older, they started living in different places, eating different things, right? Because when you're growing up, you're eating in the same home, you're having the same food, you're having the same experiences, getting exposed to the same things. And so your environment is the same. But once you get older, you start getting into a different environment. And that change in your environment and your exposure is actually going to start to alter how your genes are expressed. There's proteins that sit on your DNA called epigenetic proteins, epigenetic markers, that can change the expression of certain genes. And so, again, lifestyle experiences, cancer, depression, alcoholism, all those things can be affected by your environment. And then some traits, again, depend on multiple genes. Skin color, height, these kind of things are all what we call polygenic. And so you tend to kind of see this normal Bell distribution curve because there just tends to be this kind of natural skewing of fewer people on the extremes and more people just heterozygous blended with the different traits. All right. Thank you so much. Have a good week.